All right, so Iliana Baums speaking with us about genetics-informed strategies for effective coral conservation. Iliana is an associate professor at the Pennsylvania State University, where she studies ecological and evolutionary processes that shape coral populations to design effective conservation strategies for shallow and deep corals. She often collaborates with oceanographers to improve real-time dispersal models that help predict changes in connectivity patterns as a variability. And currently, she is particularly interested in how the propensity of corals for hybridization, asexual reproduction, phenotypic plasticity, old age, and symbiont fidelity affect their prospects for adaptation and acclimatization to climate change. Iliana is a member of the Scientific Advisory Board of Sea Corps International and the Coral Restoration Consortium. So thanks so much, Iliana, for speaking with us today.
Hello all, thank you so much for inviting me. I'm very sorry I can't be there in person, but thanks to the wonderful modern technologies, I'm able to at least share my thoughts with you on how we might use genetic tools to help coral conservation. So I would like to start with sort of giving you an overview over what my lab usually does. I work on the potential of corals to adapt to a changing environment. And I began by studying interspecies dynamics such as larval dispersal and gene flow, as well as functional versus neutral variation in shallow and deep corals. And this data is really essential when we want to predict how populations will recover from disturbances and how they may respond to selection pressures. Corals, of course, also have complex partnerships with intracellular algae, and they harbor a wild, bewildering array of bacteria and viruses. And so I have studied how symbiotic partners recognize each other, how the symbiosis is maintained, and how it breaks down. And in my view, it is definitely the case that we need to consider symbiote-host genotype interactions, as well as the interactions between the partners and the environment. And these interactions, I think, are a potential target for selection. Recently, I also began working on hybridization as a shortcut to adaptation in collaboration with Nikki Fogarty's lab. And in all of this research, I make use of a wide range of omics techniques, including genome sequencing combined with field work. And what I will do today, instead of giving you a sort of overview over my particular work, I will share with you my thoughts of how we can use all these genetic tools to help Caribbean coral restoration. I would also like to mention that I just recently gave a talk, a webinar on genetic tools. So this was a genetics 101 introduction to how some of these genetic tools work, such as microsatellite markers or SNP markers that you may have heard about. And I will not do that again today. I suggest you just watch the webinar, and it's available at this website. You see it on the screen. If you like an introduction on very basic, what's an allele, what's a genetic marker, what are the pros and cons of each. So instead, what I will do today is discuss these concepts of assisted evolution versus assisted migration, what they are and how we might apply them to Caribbean corals, and make three very specific recommendations of what I think we can do for Caribbean coral restoration. So first, I guess it's a good idea to distinguish between assisted evolution and assisted migration. During assisted evolution, what we want to do is assist corals to evolve temperature tolerance by repeated rounds of artificial selection. And in assisted evolution, we want to assist corals by moving them to new habitats that are becoming habitable because the climate is changing. And so, for example, northern Florida might be getting warm enough for corals to live there, and they might not be able to get to these new habitats on their own because of dispersal restrictions, for example. So assisted migration would be a human intervention to get these corals to these potential new habitats. So let's dig a little bit deeper into each of those. The premise for assisted evolution is that there's standing diversity within coral populations with respect to temperature tolerance. And so some individuals are able to survive hot water temperatures or resist bleaching, or are able to resist certain infectious disease outbreaks such as white band disease. And we know this is the case. We've worked on this a lot. We know in the Caribbean there's several species that do show within species diversity with respect to stress tolerance. So the procedure here really is to find those resistant colonies and breed them to produce resistant offspring, with the intended outcome that the temperature tolerance, say, or infectious disease tolerance of the population or the species is improved. The caveats are that the traits we are looking at that we want to have in these corals may not be heritable. So the reason why a particular coral is able to resist higher temperatures has not so much to do with its particular genetic makeup, but it has to do with very fine-scale microenvironmental differences, or it's because it has a certain microbiome that helps deal with these higher temperatures, and those components may not be easily passed on to the next generation. There could also be trade-offs, but we're not sure what those trade-offs may be. So if high temperature tolerance was an easy trait to select for and evolve, we would expect there to be already a selection for this, and we would have seen ongoing adaptation to these higher temperature regimes in the Caribbean. Now, we might actually have had some adaptation, but it's not yet obvious, and we're not sure at what cost those higher temperature tolerances may come. So it could be that lower temperature in return, low temperature tolerance in return may decrease, even though our recent research indicates that this is not the case for Cropper silvicornis, at least. This is work by John Parkinson. It's about to be submitted, so hopefully it'll come out sometime this year. The other aspect of this is that perhaps the trade-offs aren't so much in terms of temperature, but it may be because there are issues with other traits, such as competition with other corals, other species, or fending off corallivores, etc. Can you still hear me? I've been getting a message that the connection is difficult.
We can hear you.
Okay. So the interactions between host and simions are often not a focus of these investigations, even though I think they are important, and it could be that the traits we're trying to improve are in fact a combined trait of the holobiont, and that will make the procedures somewhat more difficult. So these are the caveats, and yet there's a lot of promise here as well. So as Dirk said, we're excited to see what the progress is in this field, and we might be able to adapt some of these procedures as well in the Caribbean. So what I think for the people that are doing sexual coral reproduction in the Caribbean, our first step is going to be to focus on maintaining genetic diversity, which is, I think, a realistic goal for anywhere in the Caribbean. And this is best achieved if every genotype that we have in our population and species contributes more or less equally to the gene pool. And because this is a way in which we know we can preserve the genetic diversity in the population. And for those of you guys who are learning in Curacao these new techniques for sexual coral reproduction, it is worth considering in your crossing protocols to make sure that you not just dump all of the gametes you collect from any of the colonies, without regards to quantities into one big cooler, but consider trying to even out the contribution of the different gamete donors in your coolers when you're mixing them together to try to enhance the possibility of preserving as much of the genetic diversity as possible. And I think the other really important aspect here is that we know for a good number of the Caribbean corals, they associate primarily with one species of Symbiodinium, and there are several genotypes of those species, of those Symbiodinium species in the corals as well, usually one genotype per colony. And so it might be time for us to put more efforts into culturing some of those really difficult to culture Caribbean Symbiodinium species that we just have not paid as much attention to. Well, I shouldn't say we haven't paid much attention to it. It's been tried a number of times, just proven to be very difficult, but I think it'll be crucial that we make some progress there as well. For assisted migration, the premise is that under climate change, habitats will shift, but these foundation species may not be able to reach the new habitat. And so the procedure would be to translocate corals or larvae to the new, as of now, empty habitats, for example, in northern Florida, or translocate corals from historically warmer sites to those that are expected to become warmer in the future. It's, say, the Red Sea to Australia, for example. Now, the intended outcome is to establish new populations in locations that enable the future survival without further intervention. So you're aiming to basically kickstart a new coral reef development in a habitat that was previously unable to support reef development. The caveats here are that such habitats may either not be known or they may not exist. And this, for example, if we stick with the northern Florida example, we know that there's a large light gradient across the Florida peninsula. So even if the temperatures become more suitable for coral reef development in northern Florida, the light levels may not be suitable for at least the current contingent or the current species of Symbiodinium that we find in these coral species. And we know that some of our Caribbean corals are very specific to the Symbiodinium species they obtain, at least under current conditions. So the experimental need here is really to find out whether we can, whether these coral species might be able to actually have a long-term stable association with Symbiodinium that can handle the much lower light levels in northern Florida. There's other areas, for example, where you might think of a more lateral translocation, say between a hot bay and a cooler bay that is not so much a latitudinal but a longitudinal gradient, and so it's more based east to west rather than north to south. That's I guess what I'm trying to say. And there it becomes really critical to think about how we are going to identify these habitats, and we just don't have a whole lot of good microenvironmental data to help us decide what those habitats may be or were really as well. Now the other issue is that if we're translocating colonies, mixing populations from for example, from not so much moving corals to new habitats, but moving corals from warmer habitats, from cooler habitats to now warmer habitats, so mixing populations, is that we could be getting outbreeding depression, and this is a topic that often is very confusing. So I'm going to give you a very, very basic sort of cartoon impression of what that may be. So Andy Chan, who's the co-author on this talk, who's a grad student in my lab, came up with a couple of very nice examples here. And so what she did here is draw a blue and a red leaf. This indicates two different habitats, in this case terrestrial. And imagine you had beetles that lived on these red and blue leaves, and there's red beetles on the red leaf and blue beetles on the blue leaf. And this just means that the beetles are very well matched to their particular micro habitat, so they're invisible to predators. Now, what happens during outbreeding depression is, say, one of the red beetles moves to a blue leaf and is able to reproduce with the local blue beetles. What you may get is a mismatch in the offspring, where the offspring, say is purple in color, and so it's mismatched to the blue leaf, but it's also mismatched actually to the red leaves. So in that case, you would, or it could be that the offspring are less fit than either parents in their local habitat. And in corals, we are just not sure whether outbreeding depression may be an issue, especially since outbreeding depression often doesn't appear until the second or third generation. And the good news is that outbreeding depression is often not an issue over a very wide range of relatedness values. So over intermediate relatedness values that you get in a regular population, you probably can transplant without worrying too much about outbreeding depression. When the populations become very differentiated, very unrelated to each other, or you even transplanting different species, you may have to worry about this outbreeding depression. So in terms of assisted migration, for those of you who are doing work in the Caribbean, I think the sort of precautionary approach is to focus on fairly small scale migration. So, migrate colonies among sites within islands rather than moving them between islands as much as possible. Now, as we're getting more desperate in having to try to rescue coral populations, you then are willing to take greater risks. So I think the first step would be to try to translocate within islands, and if there's really no other source population available, then one may reconsider and think about moving from other islands. This all requires, if we really want to do a good job on matching potential new habitats, is that we have to get good environmental monitoring going, and this is something that Rebecca just mentioned as well. And I know Andrew Baker's lab is trying to develop some models that will help us predict as well, what might be good environments to translocate into. So I think environmental monitoring is becoming a really big need, and especially on very small scales. We know the small scales matter. It could be it could be matter of meters really, or centimeters on a reef, and we just don't know how the environment is variable on those scales. There's another approach where instead of doing a large scale migration all at once, you might want to consider breeding in sort of small scale steps with locally adapted genotypes. This is more challenging with some of these corals that only spawn once a year. And again, I think we need to think about culturing Symbiodinium, because when you're translocating adult colonies especially, you're not just translocating the host, but you're also moving the Symbiodinium around. And for the Symbiodinium in the Caribbean, population structure is actually on a much smaller scale than it is on the host, so outbreeding depression might be more of an issue for the Symbiodinium than it is for the host. So finally, I want to bring this down to 3 sort of concrete recommendations for Caribbean restoration. I think we need to work on first preserving the within species diversity that we have as much as we can. We need to try to increase the number of distinct individuals that we have at each reef, because that will increase the opportunities for sexual reproduction. With that, the potential of having resistant genotypes as a result of that. And I also think that colonies should not be moved over large geographic distances, at least right now. And so let me elaborate on all of these points a little bit. How much time do I have left, Rebecca?
We have you slated until 2:50, so about 15 minutes, but you're right before the break. There's no one after you, so.
Great. No, that's wonderful. Okay, so first recommendation would be the more diversity we have, the more chances for success I think we will have for these populations. So, and this is especially true since we now have accumulated quite a bit of data that shows that coral genotypes are often incompatible with each other, so that we need to overestimate the number of diverse genotypes you need to have on a reef to enable successful sexual reproduction. And this is true for the acroporids, where there's a lot of data from the Caribbean as well as from the Pacific showing that in some species, fertilization between any two genotypes may range from near 0 to near 100%. And so, you know, this large variation in fertilization or in compatibility is a real issue if you have reefs with very few individuals. We want to have, you know, the second part of this is not just the sexual reproduction aspect, but also differential tolerance to disease and other stressors. Obviously, you have a higher chance of getting more by chance, having, you know, more tolerance to disease if there's more individuals there. So the typical example here is the Irish lumper potato, and in Ireland in the 1840s or so, they primarily grew a single clone of this Irish lumper potato. So all of the potatoes all over Ireland were equally susceptible to this blight, and because they were all clones, all clonemates of each other. And so this killed the crop and led to 1 in 8 Irish people die of starvation. This is, you know, what happens if you have a new emerging disease that hits a very genetically depauperate population. So basically, it's the difference between having identical twins on the reef versus having more diverse individuals. And having more diverse individuals just increases your chances of being able to survive future stressors. Now, for, you know, the Harry Potter example there, you know, for us it's easy to recognize that, you know, that there's twins and you know, there and not twins in a human population. However, with corals, you know, you can't just look at them and say, okay, that's a genotype, a different genotype from that one. To us, they all look the same. And so we need these genetic markers to be able to distinguish clones from each other. And for the Caribbean species, there's, you know, for most of the species that are currently a concern or on top of the list in terms of restoration efforts, we actually do have these genetic markers. For example, for the pillar coral as well as the Elkhorn coral, there's several different types of markers available, in fact. And those markers allow us to distinguish between individuals and identify these clone mates. So this does require that, you know, you need to invest in some genetic analyses of your populations to really understand what the diversity is like and how you can make use of it to increase resistance to future diseases and fertilization potential. So this brings me to the second point. The number of distinct individuals should be increased at each reef to increase the opportunity for sex. And especially in the Caribbean, we have many reefs that are dominated by a single clone. This is true for Cropper palmata. There are several reefs where we only have one silver cornus clone. Dendrogyra is particularly susceptible to this, where we have reefs that are, you know, large stands of Dendrogyra. They're all the same clone. And for these species, and this is actually also true for Orbicella fabulata, you know, a species where you not necessarily would expect this. True for Orbicella annularis, that's more expected given its sort of pillars that can easily break apart. But in all of those cases, you know, there are reefs in the Caribbean where there's very little clonal diversity, and hence, because most of these species do not self-fertilize, pretty much no chance of sexual reproduction. Even though each one of these clones may produce millions, billions of gametes each year, they may go unfertilized. So in these species, asexual fragmentation is the primary mode of reproduction. Now, if you have a situation like this where you have genotypic information and have determined that some of your reefs are very depauperate, so you only have one clone a reef, the question is, should you move a different genotype into your reef to increase the chances of sexual reproduction. So basically, should you move, adult corals within your reef tract to increase genetic diversity? And there are several considerations there. So how well can the coral survive being transplanted further away? And this could be because corals might be very locally adapted and just decrease in terms of their survival chances when you transplant them. But so far, experiences in the Florida Keys have shown that you can transplant many genotypes onto several reefs, and they seem to, at least at first sight, do okay. So they don't just all die, certainly. And hence, I think there's some good reason to believe that such transplantation may be successful. Now, it doesn't mean, though, that you transplant this coral, and even if you now have doubled the diversity from 1 clone to 2, that doesn't mean that you end up with larvae, because, as I mentioned before, a lot of these genotypes are incompatible. So it is worth, if you have that luxury, to do test crosses to make sure that you transplant the most compatible genotypes to the same areas. Now, as an aside, we just talked about outbreeding depression. Inbreeding depression is often a concern to the zoo community because they deal with mammals where inbreeding in a zoo setting is very possible if you have a limited number of animals that breed with each other. And so there are stud books developed and breeding programs that allow zoos to share animals with each other. But for these corals, especially in the Caribbean, most of these corals are not obviously inbred. And even the ones that we know to be inbred, like Fabia fragum, they don't show any inbreeding depression. This is also something we sometimes see in plants. So to me, inbreeding depression is not a major concern when we are thinking about breeding these coral populations, partly because inbreeding depression is the result of brothers and sisters breeding with each other over many generations. So even one round of mating between brothers and sisters doesn't necessarily lead to inbreeding depression, even though you might get a little bit of a higher mortality in the offspring. So inbreeding depression to me is not necessarily a concern, and it's different from the what I've just discussed before, which is the high clonality. This is asexual fragmentation. There is no sex involved at all. It's just pieces break off and grow into new colonies, and so these are identical copies of each other. That's different than brothers and sisters mating with each other. Okay, so that brings me to my last point here, and that's considering the spatial scales of assisted migration. And for that, it's really useful to have an idea of how or what the scale of gene flow is. And for this, we need to first figure out what is a population. The definition of a population is a group of interbreeding individuals of the same species that is isolated from other groups. And I like to use the example of horses that, it's just all the same species, but one herd lives in a valley separated from another herd by a tall mountain range that they just don't cross. So even though they could breed with each other, they don't because of this physical barrier. Now, in corals, physical barriers aren't as obvious. There are oceanographic barriers, and we know that those exist in the Caribbean. And Andy Chan, my student, has recently done a population genetic survey of the pillar coral Dendrogyra throughout the Florida reef tract, as well as a few other places, and found that indeed the Eastern and Western Caribbean are very much differentiated to the point where we're considering them to be, excuse me, perhaps very long separated. There might even perhaps be subspecies. So there is very little gene flow between, say, Curacao and the Turks and Caicos, or Curacao and Florida. In fact, probably there's been none over many hundreds of thousands to millions of years. In the Elkhorn Coral, I'm just giving two examples here. You know, there's now studies on many of the other species. We also actually had a coinciding break in gene flow between the Eastern and Western Caribbean that I published some time ago. But with the addition of higher resolution markers, we now know that there's actually further breaks where the Eastern and Western Caribbean are still separated, but Florida and the Bahamas are differentiated, and it might even, it looks like the Mesoamerican reef tract is also different. And so when you're thinking, you know, the Bahamas and Florida are very close to each other, yes, indeed they are, but they don't often exchange genes, and so they are differentiated populations. And so choosing the Bahamas as a source for Florida, or Florida as a source for the Bahamas may not be the best approach. The Dominican Republic and the Bahamas may be a different story. There may be more gene flow between those populations, and hence it might be a better match in helping you avoid outbreeding depression if you had to move corals over larger geographic scales. Now, and even within islands, you know, the differences in habitat are just not always obvious, especially to us. The other issue with transporting adult corals is that there's a lot that lives in the skeletons, and so we might transport unseen pathogens. And at this point, I'm wondering if we're better off trying to generate larvae ex situ and then moving the settlers rather than the adult colonies. This also has the advantage of having the juvenile corals still being relatively open to take up symbiodinium from local populations before their immune system develops and they sort of settle on their adult symbiodinium genotype. So at this point, I would recommend thinking about moving larvae if you had to move over large distances rather than adults. So in terms of assisted evolution, then the recommendations would be focusing on maintaining genetic diversity wherever possible, because of the caveats of not knowing what traits to focus on at this point and what the potential trade-offs might be. And this is best achieved if every genotype contributes more or less equally to the gene pool. We can incorporate that into our crossing protocols, and I think we need to consider culturing symbiodinium. We also have a need for molecular markers to identify and tag genotypes for both host and symbionts, and there we're making good progress. And again, I suggest you watch the Genetics 101 seminar if you would like to know more about these molecular markers and how to apply them. For assisted migration, I think our first order of business ought to be to focus on small-scale migration, to really establish better environmental monitoring, to think about using a breeding scheme where we're mixing local with pre-adapted genotypes, again to culture symbiodinium, and really to put some effort into developing large-scale, cheap molecular markers to understand population structure for many species. So finally, I want to end with this slide where, because you're in Curacao at the CCOR workshop, you're going to hear a lot about larval rearing and sexual reproduction, and you know what assisted evolution means is selection. And you know we have done temperature selection on the larvae, and we know some of those same pathways appear in the larvae that do in the adults, but the selection of adults and larvae, especially under higher temperature, may work differently because they are in very different environments, the larvae being pelagic and the adults benthic. And again, we're not sure whether selecting larvae under high temperature and having highly temperature adapted larvae necessarily translates into high temperature adapted adults. And we also need to consider the symbiodinium. So I think we have an ability to control the larvae until settlement, and if we really want to make a difference right now, I think our best bet is to put a lot of effort into getting the settlers to survive to sexual reproduction. And if we've figured out how to do this in the Caribbean, I think we're a huge step forward. And I think this is really something we we can tackle that. It's something that's doable. So I want to leave you with that, with those thoughts. And again, want to thank Andy, particularly because she had prepared an earlier version of this talk for the Florida Keys National Marine Sanctuary Scientific Advisory Board last August, and she was so kind as to share some of her slides with me. And with that, I want to actually close and take any questions if we have time. Unfortunately, I will not be able to be around for the question and answer session later on.
Fantastic. Thank you so much, Iliana. First, just to our attendees who are attending online, I want to apologize because we do now see all your questions coming in. So I've been aggregating those. There are quite a few questions actually for Dirk, and so I want to make sure we address Iliana's questions first, since she won't be able to join us during the panel discussion later. And then after, we do have a 20-minute break, so we'll start to address some of those questions that we're a bit behind on related to Dirk's presentation earlier today. So I don't see any questions related to Iliana's talk so far in the question box. Do we have any questions in the room?
Hey, Iliana, it's Tom. I just have one quick question for you. On the studies that you guys looked at for the ability of individuals to cross with each other, I was wondering whether or not you had enough genetic information on those individuals to look at relatedness or lack of relatedness, and whether or not that affected their abilities to cross with each other.
Excuse me. So we did this for a couple of the species, and we could not get, with the markers we have, within the microsatellite markers, the individuals, the crossing ability was not related to the genetic relatedness measures that we get from the microsatellite markers. But, you know, I have to caution this. This is using genetic markers to understand, so it gives you a general idea of relatedness, okay? So it gives you an idea of, you know, in a population, are there a lot of individuals that are fairly closely related, or are there a lot of individuals that are really outbred? But there's been a number of studies that have compared, if you had a captive population in a zoo, right, or in some other tagged population where you had pedigree information, so where you know this is the mom and that is the dad of this offspring, and that, you know, son or daughter produced, you know, these offspring, and they then, so if multi-generation pedigrees like that exist for a few species, and people have then used microsatellite markers to try to resurrect this pedigree just from the genetic information, and the correlation between those was very poor. So there's now new methods out that require more genome level information, and they do perform better. But if you're expecting the genetic markers to really be able to tell you down to the, you know, on the level of, you know, these are cousins or second cousins, and, you know, this is the mom and the dad, it just can't give you that at that level. So in our studies, we did not see a correlation between the genetic relatedness that the genetic markers gave us and compatibility, but I don't know that that necessarily says either way.
Okay, great, thank you. We do have a couple of questions that have been submitted online. We have one here, Iliana, that is asking, for Dendrogyra, the population in Florida is completely unique. If so, given the dramatic decline, from where would you suggest we look if we thought we needed to import individuals?
Yeah, Dendrogyra is I think our worst case scenario, because Dendrogyra is highly clonal. It has a really strange reproductive system, and it has, not only has the host very small scale population structure, it also is associated with the species of Symbiodinium that we only know from this coral. So it might have a very specialized Symbiodinium that is associated with the species called Symbiodinium dendrogyrum. So, you know, so there's, from what I can see right now, there is no good, well-matched source population. But we're at a point with Dendrogyra that I think we need to consider taking a higher risk than I would be comfortable with any of the other species, just because we're way more desperate with Dendrogyra. And so, you know, at this point, I think we need to consider trying to use cryopreservation as much as we can to cross whatever we can ex situ in Florida to generate some diverse larvae. The most diverse populations that we know of, and I think, so for example, the Virgin Islands has a ton of Dendrogyra compared to any of the recent Florida. So perhaps that would be a source, but then consider that the Virgin Island population of Dendrogyra is also not, all that abundant. So here the question in my mind becomes is whether it is because Dendrogyrum is locally abundant but overall rare in the Caribbean. Is it really worth it to transplant Dendrogyra to the Florida Keys, where we're not sure there's any chance of these corals to survive in the degraded habitats that we have? So, and it doesn't really, as far as we know, play a large ecological role in Florida. So is it really worth it to sacrifice, some of the, reduce the population size in some of those other locations to restore reefs in Florida with a high risk of those transplants dying? So I'm putting this out. I'm not saying either way. I'm just, for Dendrogyra, I'm not entirely sure translocation is going to be our best bet. But if we were to translocate, I think then we're going to have to go to some population that's not very close related, because there's just nothing there.
Okay, great. Thank you. We have a couple more here. How can you apply the ex situ farming of larvae from populations of degraded coral reefs? Usually the populations from these sites are relatively low, and there is the necessity to use other populations to increase the heterogeneity. It may be good to have an archive of populations in site, but I see this potentially being unrealistic. Do you want to comment on that, Iliana? It's more of a comment perhaps than a question.
I mean, it being unrealistic, I think yes, there's a lot of, issues with, I don't know how, but I think, sure, it's going to be difficult to get all of these crosses done, but on the other hand, just imagine, instead of having, each little outfit having $20,000 to spend each spawning season. Imagine we had $10 million, right? Those issues can be solved. They're a matter of being able to deploy simultaneous teams across the Caribbean that are monitoring at each of these sites, that are catching all of the spawning events, and that are doing the crosses. This is truly an issue. Like the collecting the spawn and making the crosses, that is an issue we can solve with money. This is not something where we have to wait for, difficult genetic studies to work out or who knows what. This is an issue that can be solved with money. So there, I think, we're really just waiting for the political will to appear, I guess, to really see this as a top priority that can be solved. So, this is logistics, I think, that we're coming against, because each of, even the smallest acropora coral still, produces a ton of gametes. So I don't know that we're limited in the number of gametes if we had enough people power to to do the collections and crosses.
Great. Okay. We also have a question about, do you think using chimerism is a good tool for enhancing genetic diversity per area in Acropora species?
So chimerism is an interesting, so chimerism refers to the appearance of adult coral colonies that look like they're one, coral colony is this continuous tissue attached with a single stem to the reef. And yet, if you use genetic tools, you might see that half of the colony is one genotype and the other half is another. And the thought is that this may be the result of two larvae settling really close to each other and growing into an adult colony, sort of sharing a skeleton, a common skeleton. This has been seen quite frequently in the Pacific, and we have, and you see it when you settle Acropora larvae in ex situ. They often do settle very close to each other, so there ought to be a potential for this to happen in Caribbean acroporids, but I have not had one instance where I have sampled an adult colony several times and have actually gotten two different genotypes. So I don't think it doesn't exist, but I think it's rather rare, at least in the Caribbean acroporids. And I'm also not entirely sure what that would gain us. We could just settle two different genotypes close together, and I mean, whether they're on the same skeleton or two different skeletons, I'm not really sure where I see the difference. Because I don't believe that they truly are mixing their genomes. So that sometimes happens in the Scythians. There are some species where that happens, where I mean, it's very cool, where like one genome will go invade another genome and basically just find their reproductive structures, and so the reproductive structures will have a different genome than the growth structures, and it's sort of like genomic parasitism. Very cool, but I don't think that is actually, I mean, I'm not aware of any evidence for that in corals, so I'm not really sure I see the difference between having two corals genotypes on different skeletons versus the same skeleton.
If you have time for a couple more, someone's asking, how do you recognize or choose resistant genotypes in the field for your assisted evolution?
Well, I think that's a really good question, and it's one that I've really been worried about because, in some cases, those are easier to recognize than others, right? So Steve Vollmer's group has, and Aaron Muller, at Mode as well, has worked on resistant genotypes against white band disease, and I know Nikki Furty has some data on this, and there are resistant genotypes of Acropora cervicornis against white band disease, and that's a genotype that you can find by, doing a fairly dangerous thing and, attaching a diseased fragment to a healthy colony and seeing if it gets infected or not, right? I mean, that's a nicely scoreable phenotype. Does the coral get sick or does it stay healthy? Our other phenotypes are they're very unsatisfactory because we've seen that, growth rates or linear extension is not a very good indicator of performance over the long run. It's really variable, and it doesn't necessarily indicate better performance because there's a trade-off with density, for example, skeletal density. And Ilse Kufner has worked on this and published on it. She has a paper in press right now at, I want to say it's MEPS, looking at what is the better trait to recognize here. So that's the one phenotype that's often measured as growth rate, or linear extension, I should be more specific. So it's linear extension, and I think it's rather unsatisfactory. What we really need is a fitness measure. But measuring fitness, especially lifetime fitness in coral colonies in the field, is very challenging and at this point not really available. to be honest, I think, again, a somewhat risky strategy, but a very quantitative phenotype is to inflict an injury of a known size on the coral colony, say, make a dime-sized plug into a skeleton and see how fast it heals over. This is a wound healing, and it can be standardized. Cheryl Woodley has worked on this as an assay potential, and I think this might actually be a fairly decent phenotype, but here's a call out to all of the folks that might be listening and looking for a thesis project. Really developing good fitness measures in corals is a real issue, and that is why I'm somewhat worried about assisted evolution as a practical approach. Not so much because I disagree with the general idea. I think that's very sound. It's the practicalities of it, and really understanding which ones are the resistant genotypes that I am not sure that we have a good handle on yet. But that doesn't mean that we shouldn't be working on it and trying to get there. Hello?
Hi, Ellie. Oh, here we go. Okay. Sorry, there was an issue with the mic. So we only have a couple more minutes left, and then we've actually eaten through our entire break. But I just want to get this question in. Someone's asking, what work is being done to discover where globally will we find the best coral habitats to perform assisted migrations? You kind of spoke to this a little bit so far, but if there's anything else that you'd like to add. Iliana?
Yeah, I'm thinking. Sorry.
Oh, no, sorry, I thought we lost you. You're allowed to think. Think all you want.
I don't know that I have a good answer to that question. I really don't know. I mean, there's a couple of places that come into mind that, are changing rapidly. I mean, the Eastern Pacific is an interesting place because it's rapidly changing, and it has a lot of environmental gradients within the region, and we know that the region is fairly isolated from the Central Pacific. Not entirely. I mean, there's gene flow, and depending on what species you're looking at, but it's fairly isolated. So the question is there a good idea of assisted migration somewhere within the Eastern Pacific? And there might be. I think there's some potential there to look into this with the appropriate sort of biophysical models, which are there's some there as well. In the Caribbean, I don't know. I mean, we have a terrible lack of fine scale environmental data in the Caribbean, and it's really unbelievable if you think about it. But we really don't know much about what our fine scale variability is in a lot of places. There's a couple of areas that have been studied well, and there's a lot of areas that are really unknown. So I'm not sure. I mean, the Florida Peninsula is attractive or the Mesoamerican Reef Tract, and so far as there, if you have a straight north-south line, you don't have to worry much about east-west migration there as well. As a test case, this might be attractive, but again, with Florida having that environment, the light gradient, which is that's clearly an issue that I'm not so sure we're going to be able to solve easily. So I don't know. It's a good question. I think that's a great topic for an intensive working group somewhere. Getting some modelers together and some restoration people and really thinking through this and seeing where we can get the best results.
Okay, great. Well, thank you so much for hanging around and answering everyone's questions. We are going to have to go ahead and move on. We have Tom Moore's talk up next. So what we can do is, if there are questions that continue to come through this afternoon for Iliana, I will kind of gather all of those together, and we can post them and hopefully touch base with Iliana, and maybe she'll be willing to answer a few more over the course of the next few days that we can post online at the forum.
Absolutely.
So thank you, Iliana, thank you so much for staying up late to talk with us today.
Oh, thank you, and I'm again sorry that I couldn't be there in person.
No worries. Okay, thanks so much. We'll talk to you soon.
Bye.